Thank you. It's a pleasure for me to be here to present on behalf of Professor Bo Chow. He had some visa problems coming through and was unable to get into the Schengen area, so he couldn't come to the meeting. The title of the talk in the program is on AI. Bo told me that this really isn't an AI talk, but it's a prelude to an AI talk. He's an expert on big data, machine learning, and AI. We just recruited him to the University of Alberta last year. He was trained in Boston and Texas. He is a computational neuroscientist and mathematician who is now completely devoted to the analysis of big data problems in mental health. The reason that Bo wanted to present these data here is that they deal with gyrification of the brain. And when we deal with neuroimaging, we do lots of structural measures looking at cortical thickness, ventricular enlargement, size of different subregions of the brain, and we use functional techniques like fMRI, resting state, and task-based, and PET scanning. But very few people look at gyrification, and it's particularly interesting because if we consider the brain and intelligence, brain size alone is not a correlative intelligence, brain folding is. As we think about the processes that occur during aging, healthy aging and abnormal aging, and in the context of psychiatric disorders, we can see that gyrification, or brain folding, may be quite an interesting measure to look at structure-function relationships. And so Bo, focusing on challenges in aging, is now planning to do more work with gyrification or brain folding. We all know that the future of the world is one dominated by the elderly, unless something very catastrophic happens. 1 in 4 people will be over the age of 65 by 2036. So 1 in 4, 25% of the world's population would be aged compared to our normal standards now. Being in my 65th year, I don't actually think that 65 is that old, but life has changed and developmental trajectories have taken a turn because of better health and environment. But 65 plus is old. Here is a cartoon for Europe and the Americas for age changes through to 2036. As things get darker, aging gets, so till 2060. It'll run through again. You can see the precipitous change in age globally. Most areas are going to be populated by the elderly. There are some young areas, mainly with indigenous people and less development, but in general, this aging trend is moving like a wave across our planet. So having good data and innovative data about brain structure and function may be very useful. And the kind of big data that Bo is playing with gyrification is exactly the kind of data that we can use for predictive work looking at brain folding changes in relation to functional capabilities of older people. So this is taken from a paper published in 2017 in Scientific Reports where a gyrification index is used using FreeSurfer and T1-weighted MRI structural images. Analysis was done of areas of inner and outer contours on the surface of the brain for the outer cortex. And using this approach, changes in gyrification have been assessed. Now, brain folding is very interesting, not very well understood. As I said before, it's correlated very well with intelligence. We know this since the work of Jerrison a few decades ago, comparing species for brain volume and cortical folding. That cortical folding correlates with intelligence. Why or how does the brain fold? The principal theories are illustrated in this slide. There's the axonal tension hypothesis that postulates that connections between proximal cortical areas will build, and increased connections will cause tension and buckling in the surface of the cortex. That's the axonal tension hypothesis. The radial expansion hypothesis says that expansion in the supragranular layer exceeds that in the intragranular expansion, and this causes buckling. And the third theory is one of differential tangential expansion, where tangential expansion across the layers of the cortex causes buckling. Three of them give rise to cortical convolution. People do not understand the basic principle, but the observation comes down to these three main possibilities. So you look at gyrification and the number of neurons. I mentioned Jerrison's work with different species. Bo has pointed out some work from 2015 by Mota and Herculano Huzel looking at number of cortical neurons for folding index and number of cortical neurons in relation to cortical surface area. And you can see different species. So you have the mouse, the monkey, the elephant, the goat, the mole, and Scandentia is a tree shrew. And in general, as folding increases, the intelligence of the species tends to increase. The elephant is a very smart creature, and that blue dot at the top is an outlier. And you can see the correlation with cortical surface area and number of cortical neurons. Surface area is all about processing capacity, of course. So looking at lifespan gyrification index in humans, Bo looked at a number of the T1 weighted MRI images, US databases, and correlated age with gyrification index. And it turns out that the level of gyrification is probably highest at around the age of 4, and then drops off in a nonlinear manner across the age span. The relationship of this to cognitive function is not known. Nobody has been able to measure it, but with this new measure of gyrification from T1 weighted MRI images, it will be possible to use this as another estimate in assessing structure-function relationships in the brains of people with increasing age. We have to be a bit careful with the data leading out beyond 60 years because the sample size drops off, but the nonlinear curve is quite robust between the ages of around 6 and 60. So you can see there's quite a precipitous drop by the age of 20 on a log scale. One of the interesting things about lifespan gyrification trajectories is that in the case of major psychiatric disorders, there are differences between these groups. And Bochow and his colleagues compared healthy controls, patients with major depressive disorder, bipolar disorder 1, and people who are diagnosed with schizophrenia. And none of these people had neurological disorders, and the normal controls were disease-free in terms of brain or metabolic problems. So you can see a small but very significant shift in two of those lines, the green one and the blue one, which are bipolar 1 and schizophrenia. Interestingly, mood disorders remains more or less on top of the healthy brain trajectory. So lower gyrification and deviated trajectories for aging in psychiatric disorders come out into this histogram that you can see. So under the age of 18, there are no significant differences. Over 18 but under 40, there's a decrease in gyrification index for bipolar 1 and schizophrenia, but no change in mood disorders. That's interesting when you think about the evidence for structural changes with mood, not necessarily emphasizing the cortex, although there's some information for anterior cingulate, but thinking about the hippocampus, for example, we do see structural brain volume changes in depression. But gyrification apparently doesn't change. And then over the age of 40, again, significant decreases in gyrification for bipolar 1 and schizophrenia. Looking at the distribution of these changes in a graph that represents the individual data points, you can see the clusters around the healthy controls in the middle in the distribution shown on the right-hand side of the graph, where it says A and B. And you can see against the central distribution of the healthy controls a shift with mood disorders and with schizophrenia and bipolar 1. This seems to have frozen. I can't. Oh, thanks. So lower gyrification in psychiatric disorders during aging. Looking at left and right hemispheres, in mood disorders there's no difference, that's clear. In bipolar 1, more changes in the left hemisphere. And in schizophrenia, bilateral changes. In this set of cohorts, more changes in the right hemisphere. And looking at left-right across healthy subjects from these three different cohorts, SA, NK1, and COBRA, there are no differences, no changes, including the lateralization analysis. So looking at this in terms of staging effect, gyrification index across these disorders, the Spearman correlation between bipolar stages and left gyrification index was significant. That's the end of the presentation. I'd just like to acknowledge Cloud. I don't have a picture of him, Bo Chow, and the collaborators. Our Edmonton group is up there on the right, and the others are from Houston in Texas at the Mental Health Center there. So thank you very much. If you have questions, I'll try and answer them on behalf of Dr. Chow.
Thank you very much, Professor Xiao.